Great. So hi, welcome everyone to the Reclones community meeting for June. Today we have a slightly longer double session with three speakers, two talk topics. So the first talk will be by Florian Hahn and Birhan Edisi on plant gene editing toolkit. And the second talk will be with I probably should check this properly. Sorry, Luis Gabriel Brieba de Castro. I hope I pronounced most of that correctly. And he'll be talking to us about an open source recombinase polymerase amplification toolkit that spans both point-of-care diagnostics and agricultural uses. So we hopefully will come away from this with a fair bit more knowledge about some open access and open enabling toolkits that we have available around the world. So without much further ado, I should just introduce our first two speakers. So we have Florian Hahn, who is currently the bioediting lead expert at GrowMore Foundation. So he studied biochemistry at the Heinrich Heine University Düsseldorf, continuing there for his PhD, where he used CRISPR-Cas9-based methods to introduce C4 photosynthesis features in seafood plants. So his specialism is in genome engineering using CRISPR-Cas technology development. And he then went to take a postdoc at Rothamsted Research and the University of Oxford, again working on editing in major crops like wheat, rice, and legumes. He subsequently branched out of academia and led the cell engineering division at the cultivated meat company, IV Farm Technologies, before joining GrowMore Foundations just over a year ago. And Florian is joined by Birhan, who is a postdoctoral fellow at the Biomanufacturing Group at ICGEB, the International Center for Genetic Engineering and Biotechnology. And both of them today will be talking about the enabled gene editing toolkit for plants. Birhan did his PhD at the University of Pretoria, focusing on agricultural biotechnology, where he focused primarily on also genetic engineering of industrial crops. He was a senior research associate at the Ethiopian Biotechnology Institute prior to joining ICGB, and has been working in this field for more than 15 years and working with scientists across Ethiopia, Hungary, Israel, South Africa, China, and Italy. So he's worked in a number of different roles within technology development and dissemination in the agricultural sector. So without much further ado, I'm just going to pass it off to Florian and Birhan to talk about more how the Enable Toolkit can be used in low resource setting countries. So just for folks who are listening again to the recording afterwards, Florian and Birhan chose not to record their talk today, but I'm sure they'll be happy to get in touch for people who want to test their Enable Toolkit. So in the meantime, we'll move on to our second talk of the day, which is with Luis. And he is currently a research professor at the National Laboratory of Genomics in Cinvestav in Mexico. He did his bachelor's in biochemistry and master's in biotechnology, both at the Monterrey Institute of Technology. He continued in the field of biochemistry with his PhD at the University of Texas Health Science Center, San Antonio, and then as a postdoc at the Harvard Medical School in Massachusetts, before returning to Mexico and progressing through various professorships at Cinvestav. He's won a number of research grants over the last 2 decades, contributed to numerous publications and book chapters, and taught many students both in his research group and on the graduate programs at the Institute. He is also honored as a member of Level 3, the highest of the National Research System in Mexico, and is committed to serve the Latin American scientific community by promoting the passion for science and creating supportive enabling infrastructure for researchers in the area. So I'm sure we'll hear more about his passion and his works on his talk on open access recombinase polymerase amplification. Over to you, Luis.
So thank you, Giankai, for the nice introduction. And thank you for everybody for listening. So as Giankai say, I'm a biochemist. My whole life I've been a biochemist. And the thing that I'm working on is on DNA replication. And basically, I just want to share this slide saying that the simplest replisomes that exist are from bacteriophage T7. Charles Richardson lab has studied that for almost 50 years. But also, I want to to put some attention that in our mitochondria and in plant mitochondria, this phage-like system exists. So I want to talk about RPA, but before that, I want to show this slide by the group of Benkovic that used to work a lot with the T4 replisome, and based on the work by Bruce Alves, and say that it's a very complex replisome. I mean, it's very complex. But one of the things that is very nice about the T4 system is that can efficient assemble homologous recombination. And that has been known for a long time. I mean, that has a system in which can you hear me well? Okay, that you can have a system in which a recombinase can use a piece of DNA to start DNA invasion. And that has been known for a long time, and as we can see in the review by Scott Morica. And almost 20 years ago, this seminal paper published in PLOS Biology by Olaf Pippenburg coined this technology called RPA that basically uses this T4 system to carry out PCR without the disadvantages of PCR that has to be to change temperatures. So, and from my point of view, when I write this paper many years later, the thing that was very nice about this particular paper is that they use molecular crowding to enhance the RPA reaction that later on seminal papers, including one by Charles Richardson, also shows that was very important. So the bad thing about this technology, RPA, is that it's used as a black box. I mean, nowadays we can do a search in PubMed, and you can see the name of several companies, or one company special that I don't think that I should name the company. And researchers uses that as a black box. I mean, the individual enzymes that are needed for RPA can be cell. I mean, but most of the researchers actually use these kits. So so we decided to try to have an open access kind of system, and we set up to actually clone those enzymes and distribute it, and we just have a paper that is almost accepted as a title of a paper in this journal that is called Peer Journal. And the strategy was very simple. I mean, for a successful RPI, we have set some genes that has to be cloned and the protein expressed. So we synthesize all those genes. I just put the USBX, and we did a simple cut and paste cloning with two enzymes that we like, that are ND1 and BAMH1, because they happen to be cheap and simple to do. And we put in a modified PET vector. The only thing that is different about this PET vector is that the cleavage site is something that is called PPS, that we can purify in-house to remove the histidine tag. And after that, we clone all the enzymes. Not all the enzymes. Some of them, as you can see, like the creatine kinase is available in that gene, and the VSU DNA polymerase is also available in that gene. So and other enzymes we just said. So some of the things that I want to say as a final point of this talk that we have to remember is that some of the tips for these reactions to be successful is to avoid glycerol. Is that in our hands, the BST DNA polymerases expresses very well. Try to express the proteins at 16 degrees using the Takara chaperone system, and use platforms for LIGO design. So this is the basic reaction of RPA, in which a single-stranded binding protein binds to a piece of single-stranded DNA. A mediator has to remove this single-stranded DNA binding protein in order for the recombinase to recombine the DNA, and a DNA polymerase has to extend for this single-stranded DNA. So in our hands, the enzymes can be purified. I mean, everything is in the paper that I hope soon will be published. So we can purify these enzymes UBX, UBY, and GP32. And the only thing that is important is that for one of those enzymes, we have to remove the histidine tag. I mean, and the yields, I mean, we can get yields that they are very high. We can get 5 or 10 mg per mil. That is mainly the yields that people need for crystallography. I'm a crystallographer by training, so I'm used to have very high yields of purified proteins. So, and the first experiments that people do, that we decided to do with our systems, is just something that is called D-loop assemble. In this experiment, we have a piece of DNA that has to assemble into a circular plasmid. So, and we noticed that we knew that in order for this to be successful, we have to have all the components. And this is different for the RPA from E. coli. It's not enough to have for RPA to have all the components of the T4 systems. We know we have to have DNA polymerases like BSU, BST, and reverse transcriptase that I showed you, and a system to generate ATP and a nuclease. But the second part of the experiment is that those primers has to be extended. So we have this primer over here that is put together in the double-stranded piece of DNA by the recombinase, and it's extended by BSU or by BST. And I just want you to notice that this band over here get lost eventually because of the highly intrinsic possessivity of the BSU in comparison with BST. So one of the things that we struggled for a bit was to have BST DNA polymerase that was soluble. I mean, we asked Professor Chaput in California for the plasmid, and in our hands, when we subcloned it with the histidine tag, was completely insoluble. We just one of my former students actually sent the plasmid to his DNA, and we subcloning, and the enzyme was perfect after and expresses very well, as you can see over here. And this is going to be in the add gene packet very soon. And obviously we have the BSU from add gene, and we have a professor from Japan was very kind to send us a version of the reverse transcriptase that is thermostable. So this is the final list of the work, but I just want to show you a little bit more, and that this is a table from the paper that somebody can actually ask for all the plasmids from add gene and do these experiments on their own without the need to do all the experiments that I will show without the need to buy any kits. So as glycerol impinges RPA, we store our proteins at 10% glycerol, very concentrated. So the final glycerol concentration in the experiment that I will show is going to be less than 0.5%. And this is very, very important because glycerol impinges the reactions that I will show. So anyways, so as you can see, we can purify and storage our proteins in a very simple buffer. This is a very simple buffer for biochemistry. And we decided to storage our proteins in a snap freezing in a fairly concentrated manner. And we have that goes to 100 micromolar. That is about 100 times the amount of enzymes that we need in the reactions that I will show you in a second. So prior to starting our RPA reactions, the first thing that we notice is that different researchers have studied this reaction, and the amount of the molar concentration that enzymes that they use, it varies a lot. I mean, you have, for example, this enzyme, the UBSY. In this particular paper, it is used at 30 micromolar, and and in the original paper by Pippenburg, is used at 1.8. So it's a lot of variation in the amount of enzymes that diverse people use for this RPA reaction. So the first experiment that we decided to do was to subclone a segment of the SARS-CoV-2 gene. So some different genes, the RNA polymerase, the E gene, the N gene, and also control the human. And obviously we can do a PCR reaction, and we can see the amplification of the bands, very small products that are often used for RPA. The only thing that this slide says is that it's working, the gene that we do. And we set up to do a nice RPA reaction with and without salt. And we can see that in the presence of salt, we have a fairly small, a fairly robust band. I think it was very nice because it was the first time that we did this particular experiment, and we used the enzyme concentration that was reported in 2006. So the next experiment was actually to try to improve the reaction. And in this experiment, we can see the PCR reaction over here in this part. And we can see that when we increase the concentration of the GP32, that is a single-stranded binding protein, we have a nice and very robust band. So obviously, if we increase the concentrations of all other proteins and keep the other two constant, we then do more combinations, because we were very excited when we saw this very robust band that actually competes with a PCR reaction. That is just to leave it like that. So the point of this slide is that we can do a PCR without a PCR machine, if you want to call it that way, just by playing with the amount of GP32 that we have, and two oligos that they are complementary to this particular region. We obviously play with some temperature, and we measure the TM of our enzymes. This particular enzyme, the UBSX and the GP32, they happen to be thermolabile. So it's not surprised that at 52 degrees, we see no reaction, because we measure this, the thermal stability by PCR and a dye, and just happen to be thermolabile. So, but we can work very nice at 42 degrees. I mean, we can, and in some cases at 47 in this particular reaction. One thing that we also did is that we add different additives like DMSO, betaine, etc., and osmolytes like tetalose or betaine, and in different to some researchers, we found that the addition of DMSO actually increases the amount of the RPA product. Yeah. And finally, not finally yet, we set up to see how many molecules can we use as a template to amplify. I mean, in the literature, I mean, they have methods using CRISPR-Cas technologies associated with RPA that they can detect 10 molecules per reaction, and that they can observe 60 molecules per reaction. In our hands, we can see for this particular gene that we can start with 1.3 molecules in the gene P, but it's different for other genes over here. So obviously, one of the caveats that we have in this particular reaction is that we let it incubate for half an hour. So, but we can see this very nice kinetic of amplification, saying that this band is actually the band that we are looking for. Yeah, so we're very happy. I mean, we can use our technology using enzymes that we purify at home and amplify from very few molecules of DNA that we purify as a template. But the point is to try to use it in field. So I did my PhD working with T7 RNA polymerase. So we put some T7 promoters in some genes over here. And the thing that you can see over here is that in a linear plasmid, because that RNA polymerase is going on and on and off and off, you have a lot of RNA versus a circular plasmid. I forgot to tell you that we also put a transcription terminator. So, and thanks for with the Harvard transcriptase published by Okano and Yasukawa, that is thermostable at 60 degrees, we can we can amplify this piece of RNA to cDNA, yeah, as you can see over here. And we also use an endophore for Thermostermophilus that we should clone over here. And in this experiment, we just use an oligo that has a 3' chain terminator and has an abasic site. So if the AP1 of the endophore clips this oligo, you can see an amplification. And that's exactly what happened. I mean, that's the way it should work. And it works, and just says, and this experiment just says that our enzyme Thermostermophilus endophore is active, recognize an abasic site, and clips a piece of double-stranded DNA and removes a 3' chain terminator. Because if we want to use this technology in a field, the way RPA works is that as we can amplify this piece of DNA, of RNA, or cDNA by 2 oligos, but very importantly, a third oligo that has an abasic site and a chain terminator has to be used to have some specificity. And when this is cleaved, you can have a small product in comparison with this one. And because we can see it over here, we have with the gene N, we have this product that corresponds to this part over here. Later on, we clip, and we have a second product that corresponds to this part over here. And we can add biotin and FAM, secondary antibodies, and have these lateral flow devices that a lot of people uses, and have clinical samples. Somebody actually gave us some RNA that we amplify to cDNA, and over here we can see this cDNA with this particular band, and we can see this amplification from clinical samples, and we can see over here that indicates that we have a clinical samples. If you have very good eyes, you can see a very faint band over here that is labeled with red in this positive clinical sample, and we can see a very strong band over here In this case over here, the same. And in negative samples, that RPA wasn't successful because it wasn't viral load over there. We don't see any band. So we have deposited all those plasmids in AdGene, just hoping that soon they will be released. I'm particularly fan of this pickled clenofragment BST DNA polymerase because it's very straightforward to purify. BST DNAP is one of the enzymes that they are very useful in biotechnology, and but it's tricky to purify. So I hope that people will start using this pickled histidine tag just to purify. I'm also hope that a lot of people will use this Endo4 with Thermophilus because this purifies extremely well. And I think that this PET19 PPS, GP32, USPY, and UBS can be used for sessions. We hope that soon the processing will be available. But the thing that is more useful, and I hope that will be more useful for people, is to pay attention to this table over here, because I know that AdGene also has some free harvest transcriptase. Obviously, I cannot put the add to AdGene this molonimo in leukemia virus by Professor Hocano, because it's but hopefully people can ask. And obviously the BSU, I'm looking for the BSU is also in AdGene. In this paper, people can find it. And very importantly, the Gabriel kinase is also available in AdGene. So, and we just add this AP1 endonuclease from Thermophilus that expresses very well and is very nice. And I hope that when people have in their mind that glycerol for some reason impinges RPA, that we try to put that very in capital letters in the paper, and people will start using and this kind of enzymes to actually do RPA without buying a black box in a kit. And I want to thank a lot of people in the lab, especially to Francisco, that this is part of his PhD thesis. As you know, this project started because of the SARS-CoV-2 COVID pandemic, and we got some funding from Mexico, and especially to Paola, Antolin, and Corina Diaz that helped Francisco cloning, people in my lab. And my apologies for go kind of fast, but I knew that we have limited time, but I'm open to questions. And I invite you to read the paper that eventually is gonna be published. It's accepted pending the AdGene point of view acceptance of the plasmids. And I will start sharing my screen, and thank you for listening.
Thank you, Luis. That was a really interesting talk, and you were bang on time. So we've got 10 minutes left for Q&As. But very interesting to see more about this open access RPA, because as you say, it's always useful to be able to, especially in various contexts, be able to create your own reaction mix that you can use, and you know what is in it, and it's not just a black box. I can see in chat that Severin, you asked a question. Would you like to ask Luis directly, or shall I read the question out?
No, go ahead, please.
Yeah, okay, sure. And I can see there's a lot of love for AdGene right now, because Severin was asking whether the plasmids that you deposited, Luis, whether they would be on the open MTA on AdGene. You're on mute, just FYI. Luis, just double checking, can you hear?
Could you repeat? Yes,
Could
you
I
repeat the
repeat?
question,
Yes.
please?
Basically, you asked whether those plasmids that you were depositing, would they be under open MTA on AdGene or just a standard UB MTA?
Yes, they are open NTA. I didn't. I mean, everything is the pattern. Yes, it's totally open.
Great, good to know. So I can see there'll be a much request from that quarter. And I'm going to jump back. Who has? Does anyone else have any questions in the meantime?
But that point that I want to make is that all the resources, because the BSU and the kinase for ATP generation are already in AdGene. So anybody can go to AdGene in a couple of months and pick up all the reagents that he or she needs.
And do you with your testing of these reagents for RPA, I mean, do you have the protocols and methods available?
Yes,
because you said you were going
to the
publish
kinase. I
them.
mean, we ask from AdGene, and the BSU we ask from AdGene. Yes.
Perfect. I see there was some discussion in chat, which I believe Florian, you've been handling. Florian, you and Eric have been handling with questions about IP. So I think Luis, you also had a question on whether the enabled toolkit will be considered for commercial use, or do you have to ask for a license?
I will need to go back to the Grow More Foundation to ask about that. So we were handling mostly the sciencey bit, but I know there were discussion around the license in the past, and I think these discussions were had, but I can't give you the specifics around that, unfortunately, no.
Not a problem. I know how tricky these kind of licensing issues can be. Lucero, you had a question? So I'll just read it from chat. So he says, thanks, Luis, for the talk. Did you evaluate activity in different batches? Because there was a significant impact on enzyme mix concentration. Let you get settled before trying to answer again.
Can you repeat the question again? Because
No problem. Yes, yeah, I saw that you were relocating. So Lucero said, thanks, Luis, for the talk. Did you evaluate activity in different batches? There was a significant impact on the enzyme mix concentration.
Yes, I mean, yes, indeed. I mean, the thing that we found is that GP2 actually, yes, and we try to we repeat that experiment with at least 4 or 5 different batches, and always was about the same. The enzymes purify very easily, and of course in the paper is going to be the protocol. Is a protocol, and it's no difference with the different batches. One of the things that we didn't try before because of time was to actually lyophilize the reagents, and that's something that I hope we can do in the future, because it's going to be, I mean, that's something that we have to do. Yeah.
I suppose lyophilizing the reagents means that you can more easily ship and distribute at ambient temperatures. That'll be more accessible, yes?
Yes, exactly. Yes, but we haven't, I mean, we did just a small try. I mean, we haven't actually been serious about doing that. Yeah. But then science purified very well, just with a simple histidine tag. Yeah.
I think there's been various attempts over from various researchers about lyophilizing DNA parts, or even trying to do this with strains, E. coli strains, so more easily shipped. So it'd be great to see all these kind of different technologies coming together to be able to more easily distribute the different reagents needed. Just going to call out again into the room if there are any more questions. I can see there's a lot of thanks and congratulations about the different presentations today. Very useful. Okay, well, if there are no more questions, I want to thank Luis, Florian, and Brieba today. It's been very interesting hearing all your different talks, and we're hoping to see all these technologies come out in the near future, so that we'll be able to more easily make use of them. Great. Well,
Thank you, Yankee. Thank you.
thanks, Brieba.
Thank you, Yankee.
Thank you,
Thanks,
Florian,
everyone.
and thank you, Liz. Well, have a lovely rest of the day, and we'll see you again next month. Take care, everyone.
Thank you.
Bye.